Hello guys, so now we will start with the systemic histology slides. In this we will cover all the systemic slides in the 9 episodes. We will study in these different systems. So for the notes you can visit my telegram channel. I will provide the link in my description or it is present in the channel bio. So now we will start with the respiratory system. In this, according to the university point of view, we have two slides important, trachea and lung. So we will start with the introduction. Now first we will see respiratory tree. So respiratory tree, as we can see here, it starts from the nasal cavity and ends at the alveoli. So we should remember this in the sequence. Nasal cavity, then pharynx, then larynx, then trachea. Trachea divides into right and left primary bronchi, then primary bronchi divided into secondary bronchi. Secondary bronchi divided into tertiary bronchi, then bronchi divides into terminal bronchiole divided into respiratory bronchiole divided into alveolar duct, which divides into alveolar sac, and lastly alveoli. Alveoli, it is the functional and structural unit of respiratory system. Now, as we can see, from the nasal cavity till the terminal bronchiole, it is called as a conducting part. And from the respiratory bronchiole till the alveoli, it is called as respiratory part. So now we will see here, as we can see, conducting part and respiratory part. Conducting part, it helps in the passage of the air. Conducting name suggests conduction. So passage of the air and helps in cooling of the air, as the environmental air is hot compared to which our body needs. So conducting part, this is a tube like structures. And respiratory part, it helps in the exchange of the gases such as oxygen and carbon dioxide at the between the alveoli and the pulmonary capillaries. So conducting part is made up from the nasal cavity till the terminal bronchiole. So includes nasal cavity, pharynx, larynx, trachea, bronchi, terminal bronchioles. Respiratory part starts from the respiratory bronchiole and ends at the alveoli. This helps in the exchange of gases. So respiratory bronchiole, alveolar duct, alveolar sac, and alveoli. Clinically, we divide respiratory tract into upper and lower respiratory tract. Upper respiratory tract includes the parts which are above the trachea, such as nose, pharynx, and larynx. We can see here diagrammatically, upper respiratory tract includes, as it is the starting point of the trachea. So above that we have larynx, pharynx, and nasal cavity. So these three comprises upper respiratory tract. Lower respiratory tract comprises trachea and parts below this. So as we can see here, we have trachea, we have primary bronchi, both the sides, then divides into secondary bronchi, then it is a lungs, and at last we will have alveoli. Respiratory system is lined by three epitheliums. Trachea is lined by pseudostratified ciliated columnar epithelium. Then next, alveoli, which is lined by simple squamous epithelium, and rest of the complete respiratory tract is lined by the simple ciliated columnar or cuboidal cells. As now we can see here, there is a single layer of cells which has a multiple layer of nucleus. So it gives a stratified appearance. Actually, it has only a single layer of cells. So it is named pseudostratified and ciliated. As we can see here, we have cilias. So it is called ciliated, and these are columnar cells. And in between this, as we can see here, white colored cells, which are nothing but a goblet cells. Now we will start with the histology of the trachea. Trachea is made up of fibroelastic cartilage, which is C-shaped. Fibroelastic cartilage is nothing but a hyaline cartilage. Why your hyaline cartilage is required? Because as when expiration is done, after expiration, the trachea may collapse, which is too dangerous. So we have here fibroelastic cartilage, which prevents collapse of the trachea. So the airway passage remains patent. Now trachea extends from the cricoid cartilage, which is at the C6 level, and bifurcates into right and left bronchi at the T4 level. Trachea is made up of four layers: mucosa, submucosa, cartilage and smooth muscle layer, and adventitia. Mucosa, it is lined by pseudostratified ciliated columnar epithelium containing goblet cells and small granule cells or Kulczynski cell. Small granule cells or Kulczynski cell, these are also called as Apud cells, help in the secretions of the certain chemicals such as serotonin. Goblet cells, the goblet cells produces mucus which traps dust particles. And lamina propria contains lymphocyte, eosinophils, plasma cells, and fibroblasts. These cells helps provides immunity against the foreign bodies, as our nasal cavity is directly exposed to the environment. So many foreign particles, microorganisms can enter the respiratory tract and cause infection. Submucosa contains connective tissue and submucosal glands. Submucosal glands, there are two types. One is serous gland, another is mucus gland. And third layer is cartilage and smooth muscle layer. Cartilage, as I said, it is a C-shaped hyaline cartilage. And smooth muscle, which is trachealis muscle. It is present posterior to the C-shaped cartilage. As we can see here, it is a C-shaped hyaline cartilage. Posterior to it, we have trachealis muscle, which is attached to the ends of the hyaline cartilage. Adventitia is made up of a fibroelastic connective tissue that contains blood vessels and the nerves. As now we will see a histology diagram. In this we can see there is a single layer of cells, but they have nucleus at the different levels. So it gives a stratification appearance, false stratification appearance. So that is why it is called a pseudo-stratified columnar, as cells are columnar. And as we can see here, cilia, so ciliated epithelium. And in between that we have goblet cells, provides traps dust particles. Cilia helps to remove the mucus. And this is the submucosa. It contains serous glands, mucus glands, and a connective tissue. And in a hyaline cartilage, here we can see hyaline cartilage, and next to it there may be a trachealis muscle. Hyaline cartilage, it will appear as a chondrocytes or a nest of cells with the background purple colored stroma. And we can see here adventitia, which is made up of a collagen fibers. Serous acini, it is made up of a pyramidal shaped cells and a round nucleus, and they appear pink colored. Mucus acini, these are columnar cells and have a flat nucleus, and they are white in color under the microscope. So the serous acini secrete watery secretion, whereas mucus acini secrete mucus, which is sticky in nature, which traps microorganisms. And hyaline cartilage is made up of a chondrocytes. Chondrocytes, these are the cells of the cartilage, are called as chondrocytes. And surrounding it, we have a white space, which is called as a lacuna. And each chondrocyte is surrounded by a stroma, which is purple colored. Under microscopic view, we can see here a hyaline cartilage. This is a hyaline cartilage. How we will identify? There will be a purple colored background, and if we zoom and see properly, we see a chondrocytes, and surrounding it we have a white space. So hyaline cartilage, we will identify cells have a white space around them, and cell size is small at the both the peripheral ends. As we can see here, at the periphery we have single cells and smaller cells. As we move towards the center, we have multiple cells which forms a nest, and the cell size is increasing. And we have a background of purple color, which is stroma. And we can see this is a submucosal layers. It contains serous glands and a mucus glands, and epithelium, which is a pseudostratified ciliated columnar epithelium. And we can see here it is a adventitia, which is made up of a fibrous tissue. For identification, we will see hyaline cartilage, then pseudostratified ciliated columnar epithelium, and submucosal glands will be seen. Now we will start with the histology of the lung. Lung, it is the principal organ for the breathing and respiration. It is present on both the sides of the thoracic cavity. Lungs are covered by the pleura. Lungs are divided into lobes. Right lung has a 3 lobes and left lung has a 2 lobe. We will remember this by remembering our right hand is a dominant hand, so it will have a more number. So right lung has a more number, yani more lobe. Left lung has a 2 lobes. Pneumocytes means they are air cells. So there are two types of pneumocytes: type 1 pneumocyte, type 2 pneumocyte. Type 1 pneumocyte, it helps in the exchange of the gases through diffusion. Type 2 pneumocytes, it helps in the production of the lung surfactant. Lung surfactants are made up of phospholipids. They prevent lung collapse. If there is a deficiency of the lung surfactant, it can cause acute respiratory distress syndrome, ARDS. Now we will see difference between the two types of pneumocytes, type 1 and type 2 pneumocytes. Type 1 pneumocytes are nothing but alveoli. Alveoli are made up of squamous cells and have a flat nucleus. Type 2 pneumocytes are cuboidal cells and their nucleus is round. Type 1 pneumocytes are abundant cells which comprises about 95%, whereas type 2 pneumocytes comprises only 5%. As now we can see here, there is a flat nucleus. As we can see here now, a flat nucleus. So these are the type 1 pneumocytes. And as we can see, this is a cuboidal cells and has a round nucleus. It is a type 2 pneumocytes, and this is a air filled space of the lung. Now we will see a diagram. As a bronchus, it is a continuation of the trachea, so it will have a similar properties as we have seen in the trachea. So as we can see here, it is a hyaline cartilage, and we have a smooth muscles, serous acini. So this all will be present. As we move downward from the trachea towards the terminal bronchioles, the size of the lumen keeps on decreasing. So it is a larger, has a larger lumen, so it is a bronchus. Smaller than that will be the terminal bronchiole. So we can describe by that. And as we can see here, the cells have flat nucleus. So these are the primary or type 1 pneumocytes, and air filled spaces are present. Now we will see histological diagram. Histological diagram, we can see here lumen, which is larger, so it is a bronchi, and smaller than that will be the bronchiole, smaller than that will be the alveolar duct. And as we can see here, we have air spaces, which is nothing but alveolar space, and we can appreciate a pink colored substance, which is nothing but of type 1 and type 2 pneumocytes. Now, identification points. Bronchi and bronchiole will be seen, alveoli are seen, air spaces are seen as a white colored area. Air spaces are white and surrounded by the alveoli. Most important identification point. So here we complete our respiratory system. In next episode we will see GIT. For notes you can visit my Telegram channel. I will provide link in the description, or it is present in the my channel's information. So now here we complete our video session. Thanks for watching. If you like the content, please like, share and subscribe to my channel for further informations. Thank you guys.